Hello, everyone. Welcome to our series, Global IMMUNOTALKs. I'm really glad to be hosting today. My name is Asya Rose. I'll be hosting today Marc Jenkins in our series. Welcome. It's great to have you here with us. As you know, there are several new updates in the program. Now the talks are also going to be streamed live on YouTube. And we are going to, and then the questions are also going to be asked there, and also in all the other platforms, only that Marc is going to answer them later on. So welcome back. It's glad to have you. It's great to have you here today. I will just introduce our speaker for today. Just to remind you that next week we also have David Whistler, who is going to talk in the Global IMMUNOTALK. So we are looking forward to see you also next week. And so briefly, I'm going to introduce Marc, and then we're going to ask him a question, a trick question. So Marc is actually a native Minnesotan. He received his bachelor degrees in microbiology from the University of Minnesota and his PhD working with Stephen Miller at Northwestern University. He did a postdoc with Ronald Schwartz at the NIH, and then he joined the faculty at Minnesota in 1988. He developed new technologies that enabled him and his colleagues to identify CD28 as a co-stimulatory receptor and understand how and where helper T cells respond to antigens in the body. He is a Pew Scholar and the recipient of multiple awards, including from the American Association, the Career Award, Excellence in Mentoring Award, and Lifetime Achievement Award. He was also the past president of the American Immunology Association, and is the member of the National Academy of Science. In his spare time, as you can see in the back of his, in his room, you can see the bicycle. He is enjoying bicycling and fishing, both in the winter and in the summer, and he is a student of the art of public speaking. So today he's going to give a talk on CD4 T cells response to food. You aren't what you eat. And we're really excited to have you today with us, Marc. Can I ask you, if you could mentor your younger self, what would you say?
I would say don't panic. And my younger self, I was a hyperbolic young man, and but I came to learn that professional setbacks are opportunities. And so in the mid 1990s, I developed a method involving adoptive transfer of TCR transgenic T cells into mice with the normally diverse T cell repertoires as a way to study the in vivo activation of naive T cells. And that method, I'm very proud of that method. It caught on. I'll bet you a beer that many of you in the audience use it to this day. But in the mid 2000s, based on work from Leo LaFrancois and confirmed by my lab, I learned that even though the purpose of this adoptive transfer method was to limit the number of transferred T cells to something more normal, we were still putting in too many cells, and that created some artifacts in terms of how well these cells were activated. That was a bitter pill for me. I felt like I had misled the field, having promoted that method so intensely. But it turned out that, and then I swore I was done transferring transgenic T cells, and I was going to find the real T cells in the real repertoire. And that created a great opportunity for me to change my approach and to develop new methods I'll share with you today. So, don't get married to your ideas. Change can be good. And I'll say again, don't panic. So
this is a great advice for every stage in your career. Thank you very much, Marc. So please share your slides, and we are looking forward to your talk.
okay, well, it's great to be here, and I really mean that, and I really commend the organizers for producing this enduring and influential venue that has really democratized scientific talks and really quite an inspirational achievement. It's also special for me today because I'm talking about my last research project as I move toward retirement. And it involves a relatively new area for me. And so I will probably make the mistakes of omission that people in a new field make. But that's what I'm going to talk today about, the CD4 T cell response to food. So I know I'm competing with other browser windows that you have open right now. So I'm going to try to give some special emphasis on reasons why this topic might actually interest you. And one is that there are new tools and perspectives coming into play in the field of the immune response to food, which is often studied under the rubric of oral tolerance. In fact, the great scholars of oral tolerance, Oliver Pabst and Alan Mowat, have recently published a review article on this field in which they use the term renaissance. And so I think this is a good time to be interested in this field. And my particular angle here is that, as I always do, I'm going to try to study this system in terms of antigen-specific T cells. And so I'm interested in CD4 T cells. So we're talking about CD4 T cells as they exist in normally diverse immune repertoires. And so that's a challenging thing because T cells in the real repertoire are rare. And so there's some special challenges to finding them. I'm also going to bring a perspective here, some conceptual ideas that have come from studies of immune responses to microbes. And instead of kind of splitting oral tolerance off into its own special world of immunity or immune responses, I want to think about it in terms of a more general conceptual model of how CD4 T cells respond to anything, and much of that informed by microbial immunology. So I think another reason to be interested in the topic is celiac disease. This has, as I'm sure as Bana Jabri told you last week, celiac disease is a global prevalence of about 1%. So it's likely that someone with celiac disease, which of course is a failure of immune response to food. And then for those of you who are interested in basic mechanisms of immune tolerance, I have something to say about T cell anergy, one of my true early loves of my career, but a kind of the poor stepchild of immune tolerance mechanisms. And I'm going to try to convince you today that finally, maybe we've found a physiological role for this mechanism of tolerance, and that has to do with tolerating food. So I want to start with the oral tolerance paradox. Oral tolerance is a very old field in cellular immunology, and it's been studied for a long time. And from the days of Landsteiner and earlier, it was known that food is a challenge to the immune system because food proteins are very different from the self proteins that the consumer is tolerant of. Their immune system is not tolerant to food proteins like from plants, which have sets of proteins that are completely different in amino acid sequence and function than any protein in a mammalian host. So there's a very good likelihood that you would have cells in your pre-immune repertoire that are not tolerant to food proteins, and yet you have to tolerate your food or you won't survive. So it's also a challenge because food proteins and peptides actually enter the mucosal secondary lymphoid organs, and I would argue the blood after meals. So this stuff gets into your body, and so it's your pre-immune repertoire is exposed or has a chance to be exposed to these foreign antigens. And again, this beautiful review from Alan and Oliver, colleagues, and it just came out in Nature Reviews Immunology, describes the many different ways that luminal contents food material can cross the mucosal epithelium, enter the underlying areas, and end up in the gut-associated lymphoid tissue where the immune system is going to have to deal with it. And so this review, I think, is very good, and it incorporates a lot of what I'm going to say today into a larger context. So I encourage you to check it out. And I would argue that immune tolerance of food must involve CD4 T cells, and I think we know this from celiac disease, which is a very highly HLA class 2 associated immunopathology. And so, of course, the role of class 2 MHC is to present peptides to CD4 T cells. So we know this failure of immune tolerance must involve CD4 T cells. And so the oral tolerance field has been, invoked the deletion of food-specific T cells, functional unresponsiveness, diversion into strange T cell subsets like TR1s or TH3s, or generation of regulatory T cells, all as mechanisms to prevent the host from pathological responses to food in the normal case. So, but we thought we had something to offer here because many of these studies of T cells, again, involved either intact TCR transgenic mice, adoptive transfer of TCR transgenic T cells, or even studies that did not involve direct access to food-specific cells. So we thought our way of going at this problem might be helpful. And so Sung Wook, when he was a postdoc in my lab, took on this project. He's now has his own lab in Korea. So just a short primer about CD4 T cells, the subject of the talk. So of course, these are cells of the adaptive immune system that use T cell receptors to recognize processed proteins bound to MHC2 molecules, which are then presented by B cells or phagocytes. And then if T cells can then engage those peptide MHC complexes with their TCRs, and then in the context of what I call signal 2, so signals through CD28, and then innate immune system cytokines, these cells that have their TCRs engaged will proliferate and then acquire various helper functions. And this, of course, has been thought of in many ways in terms of the Th1, Th2, Th17 paradigm. A more recent development came from the discovery of TFH cells, which really could not be discovered from the kind of in vitro methods that led to the discovery of these other cells. And then a twist on this, the discovery of TFHs, was the realization from work of many laboratories studying infections such as LCMV, influenza, malaria, and attenuated Listeria monocytogenes in our case, that the primary immune response by naive CD4 T cells involved this balanced production of TFH and non-TFH. And the version of non-TFH that would form would depend on the innate immune cytokines driven by the type of pathogen. So in a Listeria infection, for example, you get Th1 cells and TFH cells. And this bifurcation is driven by the activation of, in the case of the non-TFH path, the IL-2 receptor and activation of STAT5, subsequent activation of BLIMP1, and then suppression of this TFH lin. Other cells that don't get a signal to their IL-2 receptor then are free to, in the presence of IL-6 and IL-21 and cytokines like that, turn on B cell 6 and become TFH. So I think you could see the evolutionary imperative for the helper cell system to simultaneously generate myeloid cell helpers and B cell helpers for cellular and humoral immunity would be very strong. So an immune system that could do both those things, because these two systems synergize, would have a better chance of protecting the host. And I argue that this is a fundamental feature of all CD4 T cell responses. Now, if it's a helminthic infection, you'll get Th2s and TFHs. If it's a strep infection, you might get, oh, typo, old man, Th17s and TFHs, but you're always going to get TFHs and non-TFHs. And so my hypothesis for the talk today is that our immune response to food is a variant of the bifurcation model. But in this case, the IL-2 receptor STAT5 dependent limb generates peripheral Tregs that can suppress cellular immunity, and the TFH limb generates anergic T cells that are very similar to TFHs, but are arrested in their capacity to help B cells. So that's the hypothesis that I'm going to pursue today. So let's dive in. In my old talks were littered with this kind of stuff, but I can't help myself. I have to have a little Minnesota humor. So a little introduction about the method that is going to be used for all the experiments I'm going to describe today. So we are detecting peptide MHC2 specific CD4 T cells from B6 mice. And we are using peptide MHC2 tetramers, the pioneering technology of Altman and Davis. So in our case, we are expressing IA of B MHC2 molecules. These are the only class 2 molecules of B6 mice. And using the technology of Kaplan and Marek, we're linking immunogenic peptides to the beta chain, making these peptide MHC monomers in insect cells, and then decorating streptavidin and fluorochrome to make tetramers. And I'll be talking about 3 different peptides today, all foreign. In other words, B6 mice do not encode these peptides in their genomes. So a variant of a minor histocompatibility antigen discovered by Kaplan and Marek, which is called 2W. A peptide discovered by Nilab Shastri from Toxoplasma gondii that has this sequence, or a peptide that Sung Wook discovered from wheat alpha-gliadin. And so these 3 different tetramers, the red ball is either this peptide, or this peptide. So our contribution to this field was to refine the technique of magnetic enrichment to concentrate rare polyclonal antigen-specific CD4 T cells so that they were abundant enough in a sample that could be run in its entirety through a flow cytometer in a person's lifetime. So the technique is simple. Take the secondary lymphoid organs. I will abbreviate that SLOs throughout the talk, with the tetramer of interest, and then magnetic beads coated with an antibody specific for the fluorochrome part of the tetramer. Then pass all those cells over a magnetized column. And again, this can be all the secondary lymphoid organ cells from a mouse. Pass that over a magnet, in which case the tetramer bound cells will be captured. Most of the cells flow through. You can then remove the magnet and capture the bound fraction. So the cartoon is a little dramatic. It doesn't purify the tetramer binding cells. It just enriches them into a total sample of about 1 million cells that can then be probed with other antibodies. And so, tetramer-based cell enrichment is the only technique of this kind that allows you to detect the cells from the pre-immune repertoire. So here's an example using the 2W A of B tetramer, using the SLOs from B6 mice, all the SLOs from B6 mice. And if you run 1 million events after tetramer staining, but you don't enrich, the naive cells are too rare. But after enrich, you can find the naive population. And if you immunize the mouse, in this case with peptide LPS, you can detect massive clonal expansion by this method. Although for this epitope and this priming, you can detect the cells without tetramer enrichment. But in terms of immune response to food, which I'm going to show you is a very weak immune response, without tetramer enrichment, forget about finding the specific cells in SLOs. So I want to mention another advance that we've made, because tetramers can miss cells that have low affinity TCRs. So Dilipon from my lab, he used molecular evolution to produce class 2 molecules that have 3 mutations in the class 2 beta chain that allow enhanced binding to CD4. And so these kind of tetramers, and all the experiments, or most of the experiments I described today, will be involve these affinity enhanced tetramers, detect more cells than conventional cells, but still in a peptide specific fashion. Okay, so I'm going to start off with experiments in which we're gavaging peptides. This is a way that the food immunology community studies this, where a gavage needle is put into the stomach and then the peptide is delivered. You can control the dose really well. But this is not a natural thing. I get it. I understand that. And maybe I'll come back to that at the end when we talk about the immune response, just to glee it in from normal mouse chow. But anyway, so Sung looked at this experiment like several different ways. So the 3X, what I will refer to as the 3X protocol, is gavaging the peptide on day 0, 2, and 4 and assaying day 6. In some cases, also with cholera toxin, to provide a priming adjuvant so that we can model what we think is more like an immune response to a mucosal infection. And then there's the 6X protocol, which is just feeding 0, 2, 4, 6, 8, 12, and then assay 14. And so just a longer feeding protocol to ask more like what would happen with more prolonged eating of a particular food. And then we do tetra enrichment from the SLOs and ask questions about the number and phenotype of the T cells. Now, I'm going to point out everything I'm going to talk about today is from the SLOs. I understand, that's a limitation of the study. A lot of interesting action, I'm sure, is going on in the lamina propria and the interepithelial layer. That's not our strength. And I think there's a lot more that could be learned there, and other, people, and Daniel Masita and others are doing great work there, but not for me today. So here's example of these kind of data. And so in an unfed mouse after enrichment, again, you have 2W IAB tetramer binding cells. They're mostly CD44 low, as you'd expect in an animal that's never been exposed to the peptide. And I should point out that within that naive population, about 10% of the cells express FOXP3. So if you look in the thymus of a normal B6 mouse, not the SLOs, you also find amongst the CD4 single positives, about 10, you can find 2W AAB specific cells, and about 10% of those are FOXP3 positive. So we think these are thymically derived Tregs. And we find these cells in all foreign epitope specific CD4 T cell populations we've looked at to date. So how they get selected into this lineage, even though they're specific for a foreign antigen, I can't tell you, but that is a feature of the system that will come up. So after 3X feeding, there is clonal expansion and CD44 conversion. So the cells are getting activated. So in the unfed mouse, there's about 200 to 300 2W specific naive cells in the SLOs. And after 3X feeding, that goes up to about 2,000 cells. But that is very wimpy clonal expansion, because if you give an adjuvant, so you feed 3 times with cholera toxin, now you get 20,000 cells. So 10 times more clonal expansion when there's inflammation. So right away we can learn that, and no one's going to be surprised here by this, but that when you just feed an antigen without an adjuvant, this is an inflammation poor environment that's not conducive to great clonal expansion. So if you continue to feed the antigen without the adjuvant for 3 more times, the 6X protocol, the number of tetramer binding cells doesn't really change, but the fraction of the population that are Tregs goes up to about 20 to 30%. And so it's notable, I think, that after 3X feeding, the fraction of the expanded population that are Tregs is about the fraction in the pre-immune repertoire. So that's consistent with both conventional naive 2W specific cells and Tregs that are 2W specific, both expanding equally, so they're at the same percentage in the population. As time goes on, though, the Tregs become more and more abundant. And I think it's incumbent upon us to feed more and more and more and figure out when this stops changing. And just as a preview of single-cell RNA sequencing data that I'll show you, we think a lot of these cells are p-Tregs. So we were very interested in the phenotype of the conventional cells that are accumulating here. And so if you look at the tetramer binding cells and you look at the ones that are not Tregs, and you look for evidence of TFHs, most of the cells are not TFHs, most of the cells are not TH1s, are not TH17s, and using a different system, we showed they're not TH2s. So we called these, and because they're FOXP3 negative, we called these TH lineage minus cells. So because we didn't know what they were, they don't express any of the markers that we use to define the canonical lineages. So what are they? So we were very in tune with experiments done by my colleague and long-term collaborator, Dan Miller at the University of Minnesota, in which Dan had discovered a population of anergic T cells within the natural repertoire of B6 mice. So here's an experiment from Dan's paper where he gated on the CD44 high or antigen experienced CD4 T cells in a B6 mouse and displayed these two markers, CD73 and FR4. And what he found was that in the naive population that there are very few FR4 high, CD73 high cells, but in the antigen experienced population, about 20% of the cells have this phenotype. And Dan and I had done some genomics experiments, transcriptomics experiments that suggested that these genes, or the genes encoding these proteins, are abundant in anergic T cells. And Dan went on to show, using functional experiments, that these cells are unresponsive to stimulation through their TCR. So we were interested in, the question of, are these TH-lin minus cells anergic cells? And sure enough, they express large amounts of CD73 and FR4. So I will say, though, that FR4, encoded by Azumo-1R, is also abundantly expressed in TFH cells. In fact, described by Rafi Ahmed in that way. And so I'll be dancing around this issue through this talk, that anergic T cells and TFHs are closely related in some way. So just to make the point that using an in vivo system that the cells are unresponsive, so we like this peptide challenge system to look at function in vivo. And so as a positive control, this is an experiment where we've immunized with the 2W peptide and complete Freund's adjuvant. Now we're looking on the SLOs on day 8. So we enrich the cells and then do from mice that got an IV injection of 2W peptide 2 hours before. So we're re-stimulating the cells and looking at either IL-2, TNF alpha, or IL-4. And we're also looking at this TFH marker, CXCR5, to distinguish TFH from non-TFH. So if you look at the cells in this primed group, if you will, there are TFHs and there are non-TFHs. And without the peptide challenge, neither are making IL-2 at this time. But if we look 2 hours after the challenge, we can find that the non-TFHs are making IL-2, the non-TFHs are making TNF alpha, and the TFHs, some of those are making IL-4. In contrast, if we look at mice, mice that have been fed the peptide 3 times with no adjuvant, and now I do an IV challenge and look at the tetramer binders, they're very weak IL-2 producers. A smaller percentage of the population make IL-2, and they have less IL-2 per cell. The non-TFHs don't make TNF alpha, and even this very small population of CXCR5 positive cells don't make IL-4. So these cells are unresponsive, and I like the term anergic. So this is our version of the kind of experiments that Dan had done in his system. So what are these Th-lin minus cells doing? Well, and how do they relate to the Tregs? And so we did this. Sung did this experiment, which is an obvious experiment. So we got FOXP3 DTR mice, and then we treated with DT to ablate the Tregs. Then we gave the 3X protocol, and then assayed day 6. And so in the control, these are the tetrabinding cells. We think these are the Tregs. These are the expanded conventional cells, and they're not. There's very few TFHs. There's very few TH1s or TH17s, as we expect, and most of them are CD73 high, FR4 high. Now, if we ablate the Tregs and look at here after 3 feedings on day 6, the mice we know are going to get very sick from this, but they're really not very sick yet. Here are the tetrabinding cells. You can see that the Treg ablation worked, though the Tregs in the population are gone. But what's very notable is there's more like legitimate looking TFHs here, but very strikingly, there are now a lot of TH1 cells forming. So the obvious conclusion from that is that the Tregs in the system, either the antigen-specific ones or other ones from the host, are suppressing TH1 formation. And Sung looked at a pretty informative experiment here, because he then looked in the Treg depleted mice at the role of IL-2 by neutralizing IL-2, knowing that the TH1 fate is one of those IL-2 receptor STAT5 driven fates. So even when you deplete the Tregs, if you neutralize IL-2, you prevent a lot of that TH1 differentiation. And that's consistent with the idea that when the Tregs are there, they're soaking up IL-2 and they're preventing the TH1 cells from forming. So from this part, we can conclude that the absence of adjuvant effects is limiting the expansion of food-specific conventional T cells in the SLOs. The Treg cells are consuming IL-2, and that is leading to suppression of TH1 differentiation, and that many of the conventional T cells become anergic. So we're interested in digging deeper into what are these Th-lin minus cells. And I should point out, I didn't point out on the earlier slide, I forgot. We looked for markers of either TR1 cells or TH3 cells to see if those would identify the Th-lin minus population, and they did not. So we were kind of at a loss to determine what they were. So Sung looked at an experiment where he got a bunch of antibodies from BioLegend and did a so-called legend screen to look at what markers that population expressed. And he then did a principal components analysis comparing the tetramer positive Th-lin minus cells from fed mice to other populations, naive conventional T cells, Tregs from just your average B6 mouse, tetramer positive Th1s and Th17s from mice generated from adjuvant priming, the TFHs that are generated from adjuvant priming, the natural anergic cells that identified by Dan, or 2W tetramer binding cells in mice where we simply injected the 2W peptide without an adjuvant into the blood a couple times. And so I think what's pretty apparent is that many of these markers are co-expressed or have the same expression pattern in TFH the tetramer positive TH1 minus population and the naturally anergic population. The naturally anergic population and the cells generated by IV tolerization in the tetramer in the 2W repertoire are very close in terms of the markers that they express. So, that makes us think that a lot of the anergic cells that are in a natural 2W, in a natural repertoire in a B6 mouse come from sensing peptides in the blood. It also makes the point that the TH1 minus population is related to these cells in some way, although they're further in this space on this PCA plot than the IV tolerized cells are from the anergic population. And we think that's because there's more heterogeneity in here that I'll get to. But again, they're really closely related to TFH cells. So, that led to the obvious kind of an experiment at that point where we gavaged mice with two peptides, the 2W peptide and the toxopeptide. So we had more tetramer binding cells to analyze. And we did it either the 3X protocol, 3X plus cholera toxin, in which we could compare the effects of adjuvants and mucosal adjuvant, or we just continue the feeding to compare short-term feeding from a little bit long-term feeding. So then we enriched with tetramers, sorted the tetramer binding cells, or we sorted bulk CD44 low cells from B6 mice, and then followed the conventional 10x genomics workflow to encapsulate the cells, make libraries, and then analyze all the groups together in Seurat, and then we split them back out by group to see the effect of the different treatments. So our hypotheses going into this were that the 3X group, which we think of kind of as a normal priming group, will contain TFHs and non-TFH products of the bifurcation. The T-Treg cells will expand early after feeding, but P-Tregs will appear later, and that the fed-only groups will contain TFH-like anergic cells. So here are the UMAPs from that analysis, and I've set the resolution of this experiment at a level, because I learned from analyzing these kind of data that you can change the resolution, and that will determine how many clusters you get. And so I decided that I would set the resolution to the point where in the naive group, the bulk CD44 low group, Tregs and conventional cells would split out into separate clusters. And so you can see that here in the bulk group, you basically have, of the 18 clusters that are formed at this resolution, you can see that 2 of them are in the naive group, the naive conventional, and what I'm going to ID as Tregs. Now, this cluster contains proliferating cells and identified through these gene products. And in fact, we think these cells are early in the cell cycle and these cells are later in the cell cycle. Now, all these groups are pretty late in the proliferative course, I think, but what I'm going to show you is that in the 3X group, about 20% of the cells are still proliferating after 3X feedings with cholera toxin. Of course, very few are proliferating in the naive group. There is some proliferation in the 3X group. It's less than in the cholera toxin group, but the fact that the difference in the absolute number of T cells in this case is 20-fold, only might be 2-fold of that can be accounted for by proliferation. So we think there's probably quite a bit of cell death going on in this 3X group. But what is really striking to me is that in the 6X group, very, very few of the cells are cycling. And I'll show you in a minute that many of the T cells are actually becoming quiescent and taking on certain properties of memory cells. But this is striking to me because this group, remember, got fed, gavaged the peptides 2 days before this analysis. So these cells are becoming quiescent in the face of continued feeding of the antigen. And I think that in and of itself is evidence for some kind of unresponsive state being induced. So these are the other cell populations that we made calls on based on gene set enrichment analyses of various kinds. I'm not going to go through my defense of these calls at this time. It would take forever. So maybe in the discussion on YouTube, we could go into that if you'd like. But these are the calls of the non-proliferating subsets. There's 15 of those, and we could make calls on most of them. And those calls are listed here. So if we just refer first to the UMAPs, we have two different kinds of effector cells, this cluster 1 and cluster 10. They're really prominent in the 3X group and in the 3X plus CT group, and they're very not prominent in the 6X group. And the gene set enrichment we did here was based on a set of effector T cells generated in response to Listeria infection. There are then these clusters 2 and 4, which have markers of quiescence. They have many memory cell markers. I'm hesitant to call them memory cells because, of course, they're staying quiescent in the face of continuous antigen feeding. So I'm just calling them quiescent. So they're here, and you can really see them accumulating in the 6X group. There are Th17 cells here. There are Th1 cells here. There are TFH cells here. And then there are the TTregs that you can actually see here in the 3X and 6X groups. And they are, and then there are also PTregs that are quite far in UMAP space from what we think are the TTregs, and then what we think are XTregs here in this cluster 15. And so there's a lot of this explosive diversification of the cells in the pre-immune repertoire into forming all these different kinds of cells. And so what about our hypotheses? So and these are these stacked bar graphs where the percentage of all these clusters as a function of the number of cells in each group are being shown. And so I'm going to highlight the boxes of interest. So these are the Th17, the Th1, and the TFH cluster. And these cells form well in the group that's fed 3X with cholera toxin. These cells are not prevalent in the groups that are fed with peptide alone. So the conventional canonical kinds of helper cell subsets that we think participate in immunity are not forming well with antigen alone feeding. And I would even argue that these cells in the 6X fed group, which fall within cluster 6 at this resolution, are really falling, on a very kind of edge of cluster 6. So are these really Th17 cells, or are they something, closely related, but an unresponsive version of Th17 cells? And the same can be said here for the TFHs that are in cluster 3 in the 6X group. And I think you'd agree with me, they sit in a different part of the UMAP space of cluster 3 than the bona fide TFHs. So canonical T cells are not, T cell subsets that have participated in immunity are not forming. Also, again, these clusters 1 and 2, these effector clusters, they're prominent in the 3X group. They're not prominent in the 6X group. And vice versa, these quiescent cells, clusters 2 and 4, they're not very prominent in the 3X group, and they become prominent in the 6X group. So between the 3X and the 6X feedings, the effector cells are going away, probably contracting as in all responses, and they're being replaced by some kind of undifferentiated quiescent population in the 6X group. Again, it's remarkable to me. These cells are becoming quiescent, re-expressing IL-7 receptor in the face of continued feeding. And so I think that implies some kind of energy. I would note that the TT regs are about the same percentage, they're 10% of all of the cells in all the groups. And so we think that's consistent with the TT regs that are in the pre-immune repertoires of the toxo and 2W specific populations are expanding at first. The only group that really has a lot of PT regs are the 6X fed group. So these cells do accumulate with progressive feeding. And the appearance of those PT regs corresponds with the appearance of this population, which sits in UMAP space next to the PT regs and actually close to Th1 cells, which of course don't form in the 6X group. So we have Th1-like PT regs when we think they are throwing off Th1-like XT regs. Again, Dan Miller has done some interesting gene set enrichment analysis to support that conclusion, although I guess I would call that a tentative call at this point. And then if we look at these TFH-like energic cells, so clusters 5 and 8, which sit very close in UMAP space to real TFHs, but these cells don't express CXCR5. They are more prevalent with feeding alone, although they do form in the adjuvanted group. So it's not like the adjuvant is completely preventing their production, but when the adjuvant is there, it's almost as if they could progress further to the full TFH function, whereas without the adjuvant, they stall, don't get there, or if they do progress, they're in this strange kind of TFH limbo land. So we think that you're not getting conventional canonical helper cell subset differentiation going on here. So what you're left with in the 6X group really are some cells that are quiescent and have features of memory cells, including IL-7 receptor expression, a bunch of T regs of both the TT reg and PT reg variety, and maybe some XT regs, and then a bunch of these energic-like cells. So the prediction is that, well, the reality is the Th1 minus population is a lot more complicated than we thought, and the conventional members of that population should have at least two kinds of subset. These quiescent memory-like cells and these more energic-like TFH-like cells. So we looked for that by flow cytometry. And so here again is our work, is our gating scheme on 2W tetramer binding cells in mice that got the peptide 3 times. And so, about 10% are Tregs. Again, we think these are TT regs at this time. We have the conventional cells, very few TFHs. We look at those, and most of them have this FR4 CD73 high phenotype. But if we display FR4, again, an energy marker versus IL-7 receptor, we get this kind of amorphous blob here. Although you can kind of get the feeling that maybe these cells are splitting along this axis. And the TFH energic-like ones have a lot more CTLA-4 than these, what are mainly in that undifferentiated effector clusters, cluster 1 and 10. These cells also have CTLA-4, but they have less, a lot more than a naive cell. So by 6X feeding now, we get more Tregs. Again, we think that's the addition of PT regs coming from some conventional source. Still, the cells are all FR4 high, CD73 high, but now we're getting clear formation of cells that have a lot of FR4 and not much IL-7 receptor, and cells that have the opposite phenotype. So we can document by flow cytometry at least this additional heterogeneity in the Th-lin minus population. TFH-like anergic, and then this undifferentiated, we think also anergic population, because again, they're not responding to the additional feedings. And re-expression of IL-7 receptor is good evidence of that. They're tolerized to the point where they don't know the antigen's there anymore. So they do what T cells do, and they convert to becoming more like a memory cell. So one more slide before I can conclude here. And that relates to these models, which, I don't love. They involve peptides. Most of our food is not in the form of already processed peptides. And this gavage thing, I really don't want anyone to gavage food into my stomach. So I'd like to get away with this and go to a more natural system. And so I just want to end the data part here. I really like this experiment, and it takes advantage of that gliadin, alpha gliadin peptide IAB epitope, that another old guy typo there, tetramer that epitope that Sung Wook made, and that tetramer that Dilapon made. And so this lets us go into any old B6 mouse that is eating mouse chow, which is number one ingredient in mouse chow, I guarantee in your facility, is wheat products. That's also true from Jackson Lab. So any mouse you order from Jackson Lab has been eating wheat since it was weaned. So I want to show you an experiment from the SLOs that should be unmanipulated 7-week-old B6 mouse. It means it's never been exposed to 2W peptide, but it's been eating mouse chow, standard mouse chow, since it was weaned. So if we say the mouse is weaned at 3 weeks, it's been eating the mouse chow for about 4 weeks. And so I did, and I actually did this experiment myself. I did a tetramer enrichment from the SLOs. I did a double tetramer enrichment with 2W, to which the mouse has never been exposed, 2W IAB, and the gliadin tetramer. And again, the mouse has been eating mouse chow for 4 weeks. So if we just look at the 2W population, again, as I've shown you before, most of the conventional cells are CD44 low. There's a few Tregs in the population. They also tend to be CD44 low-ish, although a little higher. And the conventionals are not FR4 high, CD73 high. No surprise there. But if we look at the population of gliadin-specific cells, and a mouse has been eating wheat for 4 weeks, number one, there's 3 times more cells. So we think there's about 300 2W-specific cells per mouse. So that means there's about 1,000 of these now in this mouse. And if we look at the phenotype, there's at least 3 kinds of cells here. One, there are cells that are CD44 low, FOXP3 negative. So they look a lot like the cells in a 2W specific mouse. And in fact, they're not FR4 high, CD73 high. What's interesting about these cells is they stain less well with the tetramer. So these might be low affinity cells, and at the level of antigen presentation going on in the gut, they just don't get activated. However, there are 2 differences from the 2W repertoire here, and one is there's a lot of Tregs, and 50% of the cells in this mouse are Tregs. And in some mice, we look at 70% are Tregs. So you get a lot more Tregs generated from this natural system where they're just eating the food every day than you would even from the 6X gavage system. There are also CD44 high cells in this repertoire. So these are antigen experienced cells. And again, they tend to bind more tetramer than this population. So these probably have a higher affinity TCRs. If you look in this population, most of the cells are FR4 high, CD73 high, but not all are. And these might be these more quiescent memory-like cells. So we like this model, and as a way to go to a more natural way to study the CD4 response to food. So to end, I would argue that when food is eaten There are antigen-presenting cells in the gut, SLOs, that are not experiencing an inflammatory situation. And the ones that have food-specific TCRs then start to respond, both the conventional type and the Tregs that are in that repertoire. And then these early undifferentiated effector, conventional effector cells undergo the bifurcation. And so if they get a signal through IL-2 receptor, they will become Th1 like P-Tregs, and some of those destabilize and become Th1 like X-Tregs. But there are other cells that don't get that IL-2 receptor signal, and therefore they're not prevented from at least starting to become like a TFH cell. And because the gut is an IL-6, IL-21 poor environment, they also don't become real TFHs. So they end up in a context of continuous TCR signaling in this TFH energic-like subset. But there are other cells that are following a different path. They really don't have TFH or non-TFH like cell markers. They take on some aspects of memory cells that are also forming. So with the 3X group, we're mainly getting these three kinds of cells. And then as time goes on, we get all these kinds of cells. And in a natural food specific repertoire, we are getting a lot of cells that have the Treg phenotype, and 2/3 of them at least are like that. The other third that actually have a good enough TCRs to respond are more in these two categories. So lots to do. And again, I didn't say anything about disease. These mice are completely healthy, and they don't have IL-15 in their gut, like things that Bana was talking about. So we hope to find a way to make them sick so that we can study that. We'll do more experiments with the natural feeding model. And when I think back to Dan Miller's results in that 20% of CD4 high conventional cells that are in every B6 mouse, I'd like to think they got that way by responding to food peptides in their blood. And so that's my story. Thank you for listening. I think I have figured out how to join the YouTube discussion for questions later. I welcome those questions. I thank the organizers for giving me this great opportunity. And I thank my students and postdocs who have helped me obviously do this kind of work. So thanks everybody, and I'll leave you with my final thought: don't panic.
Thank you very much. It was fascinating, and thank you for sharing it with us. As you also can see in the chat, now we have the questions, and Marc will be able to answer them just post them on the YouTube channel, and he will be able to answer them there. And you can also see them on X and on the other options. So you have all the information here in the chat, and we hope to see you next week. So thank you very much, Marc, again, and looking forward to meeting in real life. Bye, everyone.
Bye-bye. Thank you.